have our speakers sit here during the rest of the presentations. We want to keep Jeffrey company. Well, okay, we can do it either way. Our next speaker is Dan Everitt from TB Alliance.
Thanks, Carl, and good morning. In a few minutes, I'll give some highlights of the results of our later stage clinical trials of new regimens. But first, I'd like to share a little bit of our enthusiasm about the pipeline of drugs we have in the discovery and the preclinical development space. There we go. That's a little bit hard to see, but this is our pipeline slide. It's highly collaborative with many other partners. This is available on our website. But what I'd like to do, I mean, clearly what we need for the future are new drugs with new mechanisms of action to be able to combine together to, again, develop more than one of the sort of the holy grail, the universal pan-TB regimen that won't require any drug susceptibility testing, at least initially. In preclinical development, so these are the drugs that are in toxicology testing right now and will be moving into humans before too long. I'll just say a word about three of them. First, we do have an oral DPRE1 inhibitor that will be going into humans for the first time later this year with an IND package. I don't intend for you to be able to read all the details of the others, but let me touch on two others that are moving along very quickly, are in toxicology studies, and are expected to have IND submissions and first-in-human studies into next year. The first is TBA-J. So J is for the collaboration with Janssen. It's a second-generation bedaquiline, essentially, a diaryl quinoline that aims to address some of the aspects that are maybe not as favorable as we would like with bedaquiline, sort of a follow-on. So in vitro and in vivo, it's more potent than bedaquiline against TB clinical isolates. The MICs are lower. And then in the bulb-C mice study, it's actually shown superior shortening of treatment when combined with pritominid and linezolid. I'm not going to go into all the details. This is sort of a fast overview. A key aspect, though, is that it has a much higher predicted clearance and thus shorter half-life and less tissue distribution in humans than does bedaquiline. And from a safety standpoint, it has a much less potential to inhibit hERG channels and to prolong QT intervals. So again, it addresses several issues to provide as a follow-on, a backup second generation to bedaquiline. The other category we've been working on with a lot of promise is to come up with an oxazolidinone that's safer than linezolid and offers an alternative in addition to sutazolid that we hope will be back into development before too long. And this is TBI-223. It has very similar potency to linezolid in vitro and in animal models, in the bulb-C mice model. It's a little hard to see. The graph over on the right shows at 4 weeks and at 8 weeks reductions in CFU counts in the lungs of mice treated. And what you can see is at 8 weeks, the lowest graphs there, first are with linezolid combined with J stands for Janssen. It's really bedaquiline and pritominid. And then over on the right side is the new TBI-223 gives very similar effects of almost completely eradicating in 8 weeks all colony-forming units in mice infected. From a toxicity standpoint, again, the aim has been to reduce or completely eliminate the myelotoxicity and the neurotoxicity seen with linezolid that's well known, to reduce the the inhibition of the mitochondrial protein synthesis, which is thought to be the genesis of the toxicity. And here is just a little bit, if you look at the MPS, this is the inhibitory concentration, the IC50 in micromoles of the mitochondrial protein synthesis. Linezolid IC50 is about 8.5, TBI 223, it's over 74. It's not present at the highest tested. And if you look a couple of rows down, 28-day rat studies have been done, which are enough to show at 150 milligrams per kilogram, decreased reticulocyte counts, platelet counts, bone marrow toxicity, and that's not been seen at the highest dose tested of 300 milligrams per kilogram of the new compound. Similarly, dogs, 14 days, it's a shorter period, but toxicity is seen at the 14 days, above the no adverse effect level of linezolid of 20 milligrams per kilogram, but it's not been seen, TBI 223. So that's moving forward also with an aim for an IND filing next year. Let me move from that to our clinical trials. You've heard some comment about the NIX-TB trial. We did present this at the union meeting, but I have an update for you on it. So this is our trial of pratominid, bedaquiline, and linezolid in patients with XDR-TB or patients who have failed MDR-TB or are intolerant, are basically getting deaf on injectables with MDR treatment. So just the basic design, it's open label, it's not controlled. These are largely XDR patients who, before the brand new drugs came into being, had about an 80% or higher mortality rate when followed over 5 years. Very, very sick group. And patients coming into this trial, many of them have been already treated for months to years for MDR or XDR-TB already. So they received 200 milligrams of pratominid, the standard dosing scheme of bedaquiline, 400 milligrams daily for 14 days, followed by 200 milligrams 3 times a week. And then linezolid, we are starting out with a 1,200 milligram, the full dose. Investigators have the ability to reduce the dose to 600 or 300 milligrams a day, or to interrupt the dose for toxicities and then resume it later on. Treatment 6 months of the 3 oral drugs. If somebody is not doing well clinically and has positive cultures, the last positive culture at 6 months, they can go on for 3 more months of treatment. The primary endpoint is 6 months after the end of therapy, but we are following them up for a full 24 months after the end of therapy. So to date, we have 75 patients enrolled across the 2 sites in South Africa. We have just opened the 3rd site in Durban and are shortly going to open a site in Tbilisi, Georgia. 31 patients have reached 6 months since completion of treatment. We actually have over 40 patients that have completed the full 6 months of therapy. No patient has been culture positive at 4 months and required or went on to an additional 3 months of therapy. So these 31 patients, we've had 1 relapse, 1 patient with XDR at 3 months after completion of therapy, had new positive cultures, symptoms of TB, is being retreated with a broader spectrum, broader regimen. We don't have whole genome sequencing yet to know whether that's a new infection or a relapse. We're assuming it's a relapse. It is XDR. In addition, 4 patients in 2015, very early in the study, within the first 7 weeks of treatment, died. 1 had a GI bleed, 3 had, on autopsy, multi-organ TB, were very sick. They had very low body mass indices. 1 was about 12, low albumins, but no deaths since then in the last year and a half. All surviving patients have been culture negative at 4 months, and about 75% were negative at 8 weeks. as it stands. So we continue to enroll in that study. We have planned and have just submitted to the MCC in South Africa the next study, our NC007 study, which has been branded now as the XENIX study, which, and just so you know, the NIX-TB study, we will continue enrolling at sites until the XENIX NC007 study is launched. Then we will stop enrollment in the NIX-TB study. So the XENIX NC007 study really aims to both replicate what we've seen in NIX, but also to learn more and to optimize the linezolid dosing. So rather than starting everybody at 1,200 milligrams across the bat, we're going to look at both different durations and different doses of linezolid. And this is the overall study design. Patients will be randomized. It will be double-blind to linezolid, dose and duration, obviously open label to pritominid and bedaquiline. Bedaquilin's being given actually at a daily dosing regimen of 200 milligrams a day for the first 9 weeks, actually, then followed by 100 milligrams a day, which will approximate the same exposures that the registered dose gives, but it will be a dosing regimen that we hope will be easier in the future. As you can see, and pritominid is given at 200 milligrams a day. We're enrolling actually 30 XDR patients per group. So there will be a minimum of 120 XDR patients and then an additional 60 patients who are pre-XDR or MDR treatment failures. So one group will start out at the 1,200 milligrams of linezolid daily for 6 months. One will get 1,200 milligrams of linezolid for 2 months. One will get 600 milligrams of linezolid for 6 months, and one will get 600 milligrams of linezolid for 2 months. Same arrangements if there is toxicity of concern, and we do have guidelines for changing doses. The investigators can pause and then restart the linezolid or drop the dose. But again, it will be blinded. 6 months of treatment, same arrangement. An additional 3 months can be given if somebody is culture positive at the end of the 6-month period, based on probably the 4-month culture. I'm going to leave it at that. NC005, just one slide on this. This was our study which we presented a few times. That is the bedaquiline, pritominid, pyrazinamide, moxifloxacin, or the BPAZM regimen. And the brief design of that, drug-susceptible patients were randomized either to BPAZ and either bedaquiline at the standard licensed dosing regimen or at the 200 milligrams per day dosing, or to Rifafor, the HRZE brand. MDR patients were given the same regimen, but with moxifloxacin added, so that if they turned out to be pyrazinamide resistant, they didn't have to be taken out of the trial. Everybody was tested by line probe assay to be sure they're fluoroquinolone susceptible at the beginning. It was an 8-week study for the primary analysis. They are being followed up for mortality and serious adverse events for 2 full years. And the primary endpoint was based on 16-hour pooled sputum samples. And I'm just going to show one slide of the results. This is the binary liquid and solid culture that we talked about yesterday. If you just look at the liquid culture overnight sputum, which would be the most sensitive to detecting any bacteria, again, look at the bottom, the HRZE control, only 51% were culture negative at 2 months, whereas in the BPAMZ, the MDR patients who are pyrazinamide susceptible, 96% were culture negative. In the pyrazinamide resistant patients, 78% were culture negative. So we were, very enthusiastic about this and are currently in the process of planning the next steps for the next trial of the BPAMZ regimen. So just to finish off, what we're looking at is these two regimens combined could treat all patients with TB. The patients with XDR or pre-XDR could be treated with the BPA-L regimen, and again, we are aiming to determine, a better way of dosing L to give better instructions. Those with either MDR or drug-susceptible TB could be treated with the BPA-MZ regimen once a registration trial is done. The hope is that the BPA-MZ regimen would be a 3 to 4 month regimen for drug-susceptible patients anyway. And finally, the sort of treatment, the diagnostic algorithm is fairly straightforward. Patients could be either evaluated with GeneXpert, or in places where there's no drug resistance, they could just be treated with BPA-MZ. If they're RIF resistant, then a rapid quinolone test should be done. If they're quinolone susceptible, they can be given the BPA-MZ. If they're resistant, they could go on to the BPA-L. And that's the paradigm we're taking forward. So I'll just finish with that. I'd like to thank everybody who's made it possible, particularly the trial participants who have enrolled and entrusted their health to our investigators and us. Thank you. So, any clarifying questions?
Thank you for that. I was wondering, are there any studies that you can share with us, any plans for working with sutazolid?
Working with sutazolid, I just say at the TB Alliance, we're sort of encouraging it to get back into development, and anything we can do to help that, we're happy to, but it's about all I can say for right now. I was pleased to hear that Orem Institute is, trying to work with its development as well.
Hi, thank you. Certainly very encouraged by the NICS results. I'm curious with the high dose, how frequently are you seeing neuropathic side effects within your study population? Because I haven't seen that as much.
Yeah, I didn't show the results of that. So the toxicity with linezolid, the majority of patients do have interruptions. A few have made it on 1,200 milligrams all the way through. The minimum amount of dosing that anyone's gotten actually is 4 months worth with some interruptions. So the investigators have kept people on. I don't have the exact figures off my head, but I'd say ballpark sort of 20% of patients have had interruptions for neuropathic side effects. We've had no retinal effects at all. For the myelosuppression, everybody who's had an interruption for myelosuppression, it's usually been anemia, has been able to go back on a lower dose and continue all the way through. A small number of patients with severe neuropathic pain at 4 or 5 months have discontinued completely.
Thank you.
Wim?
Yes, Dan, given the promising results, any plans for pre-approval access to Protonamid, compassionate use, expanded access plans?
Yes, we're currently really working actively, and a couple of my colleagues in the room here are sort of assigned with leading that task of looking into a compassionate use early access. Developing that, we're also needing to look for funding to support that, but to allow Protonamid to be used outside of the clinical trial setting. So it's in active development, but the timing's not entirely clear right now.
Thanks.
Thanks.